Hi, I'm Dr. James Hutcherson, and this is a presentation on the nervous system, part 1, basic structure and function. So general characteristics of the nervous system. So it's the master regulatory system. It sends and receives information. Sensory input includes detection of changes, integration and processing, which has to do with making decisions. Motor output, which stimulates muscles and glands to respond. It also maintains homeostasis, and it acts as a center for thought, learning, and memory. The main cell types of the nervous system include the neurons, or the nerve cells, which respond quickly to changes and stimuli. It conducts electrical impulses via neurotransmitters. And then the neuroglia, which protects, supports, insulates, and nourishes neurons. They do not conduct electrical impulses like the neurons do. This shows the flow of information in the nervous system from sensory input to the brain and spinal cord for integration, and then motor output to what we call the effector, which is the thing that does something. We divide the nervous system into the central and peripheral nervous system to begin with. The central nervous system includes the brain and spinal cord. The peripheral nervous system includes all of the other nerves, basically cranial nerves and spinal nerves. So it connects the central nervous system to the other body parts. And there's two subdivisions, the afferent, also called the sensory, and the efferent, which is the motor. The sensory division has sensory receptors which perform sensory function, like detecting the changes. Receptors convert information into impulses, and impulses are conducted along the peripheral nerves to the central nervous system for integration. The motor division includes neurons that transmits impulses from the central nervous system to the effectors, and they perform motor function. Effectors are muscles or glands that are outside the nervous system. There are two subdivisions of the motor division, the somatic and the autonomic. The somatic transmits voluntary commands to skeletal muscles. The autonomic transmits involuntary commands to viscera, or organs. Figure 10.2 shows you these divisions of the nervous system. Okay, central and peripheral, and then peripheral being split into sensory and motor. Clinical application, migraines. These affect about 12% of the US population. Signs include pounding head, nausea, aura, which is a shimmering image in the visual field, and light or sound sensitivity. Environmental triggers include bright light, certain foods, lack of sleep, stress, high altitude, stormy weather, and excess caffeine or alcohol. Hormonal triggers in women may cause migraines just prior to menstruation. This lasts 4 to 72 hours usually. There's a period of excitation followed by unresponsiveness in particular neurons, stimulating the production of pain sensations in the areas at the base of the brain. A good first approach is to identify the triggers and then to avoid the triggers. Drugs called triptans may stop migraine attacks. Some triptans constrict more blood vessels than others, which is risky to some patients. Transcranial magnetic stimulation also works for both acute and chronic migraine. Treatments to lower frequency of chronic migraine attacks include injections of the botulinum toxin or Botox, and a drug that binds a neurotransmitter that causes dilation and inflammation of blood vessels associated with migraine. Some drugs have been repurposed to decrease the number of migraines, such as some antidepressants, anticonvulsants, and drugs used to treat high blood pressure. So we said neurons were kind of the main cell that actually transfers the messages, and they vary in the size and shape. They may differ in length, number, and the size of axons and dendrites. They share certain structural features, though. There's a cell body, which is also called the soma, or perikaryon. It contains a nucleus, cytoplasm, organelles, neurofilaments, chromatophilic substances, also called Nissl bodies. The dendrites are the branched receptive surfaces, so a neuron could have several of these coming into it. An axon transmits the impulses and releases neurotransmitters to another neuron or effector. And this could be another neuron, a muscle cell, or a gland cell. And a neuron may have only one axon. Structural features of the axons include an axon hillock, which is a cone-shaped area of the cell body from which the axon arises, and collaterals, which are branches from the axon terminals, which are specialized endings of extensions from the axons, and a synaptic knob, which is a rounded ending of a synaptic terminal. There are also Schwann cells that surround the axons. These are neuroglia of the peripheral nervous system that wrap around some axons in layers. Myelin is a mixture of fats and proteins that fill the layers made by Schwann cell membranes. Myelin sheath is a wrapped coating around some of the peripheral nervous system axons, and it's composed of layers of Schwann cell membranes and myelin and acts as an electrical insulator. Nodes of Ranvier are gaps in the myelin sheath between the Schwann cells. This figure shows you a common neuron with dendrites, the body, axon, and axon terminals, and the myelin sheath, nodes of Ranvier, and the Schwann cells. Figure 10.4 shows you how myelination of the axons occurs. Not all axons are myelinated. So the myelinated axons are coated by a myelin sheath, and it's produced by a series of Schwann cells lined up along the axon in the peripheral nervous system. It's produced by oligodendrocytes in the central nervous system. Groups of myelinated axons in the central nervous system comprise white matter. It increases conduction speed for electrical impulses. Unmyelinated axons are encased by Schwann cell cytoplasm in the peripheral nervous system, but there's no wrapped coating of myelin surrounding the axons. Groups of unmyelinated axons in the central nervous system comprise the gray matter. Figure 10.5 shows you an actual slide of myelinated and unmyelinated axons. Multiple sclerosis is the destruction of myelin sheaths in the central nervous system by an immune response. Myelin is attacked by a person's own antibodies. Scars or scleroses are left behind, which stop neurons from conducting impulses. Without input from motor neurons, muscles stop contracting and undergo atrophy. Other symptoms include fatigue, mood problems, blurred vision, and weak, numb limbs. Treatments involve drugs that suppress the immune activity. Classification of neurons by structure include multipolar neurons, bipolar neurons, and unipolar neurons. Multipolar neurons have many processes that extend from the cell body. Many dendrites and one axon. 99% of your neurons are multipolar. Most neurons of the central nervous system are this type. Some are in the autonomic nervous system. Bipolar neurons are composed of two processes that extend from the cell body. So like one dendrite and one axon. These are not that common, but you find them in the eyes, ears, and nose. Unipolar or pseudo-unipolar neurons have one process that extends from the cell body. Two branches that function as one axon, a peripheral and a central process. Cell bodies are mainly found in the ganglia of the peripheral nervous system. Figure 10.6 shows you the different types of neurons. We can also classify neurons by their function. Sensory or afferent neurons carry impulses from the periphery to the central nervous system, which is the brain or spinal cord. At the distal ends, it contains sensory receptors to detect changes, and most are unipolar, some are bipolar. Interneurons, or association or internuncial neurons, link neurons in the central nervous system. They relay information from one part of the central nervous system to another. They're multipolar, and some cell bodies cluster to form nuclei in the central nervous system. Motor or efferent neurons carry impulses from the central nervous system to the effectors. So these are muscles or glands it carries them to. They're multipolar. And some somatic neurons, they control voluntary skeletal muscles. In the autonomic nervous system part, it controls involuntary smooth and cardiac muscles and glands. Figure 10.7 shows you the functional classifications of the neurons. So the sensory, the interneurons, and the effectors. And as we said, you also have neuroglia. These provide structural support for the neurons. In the embryo, they guide the neurons into position, and they might stimulate specialization. They produce growth factors to nourish neurons, and they remove excess ions and neurotransmitters. They aid in the formation of synapses. Astrocytes are specific neuroglia. They connect neurons to blood vessels, exchanging nutrients and growth factors. They form scar tissue. They aid metabolism of certain substances. They regulate ion concentrations, such as potassium. They are part of the blood-brain barrier. Oligodendrocytes myelinate central nervous system axons and also provide structural support. Microglia are phagocytic cells, and they provide structural support as well. Ependymal cells line the central canal of spinal cord and ventricles of the brain, and they cover the choroid plexuses. They help regulate composition of cerebrospinal fluid. The ciliated cuboidal or columnar cells are a part of these. This figure shows you the different neuroglia of the central nervous system. This figure shows you falsely colored scanning electron micrographs of a neuron and associated neuroglia. Okay, some other neuroglia, these specifically of the peripheral nervous system. The Schwann cells, they produce myelin sheaths that are found in some of the peripheral axons. They speed up the nerve impulses. Satellite cells support clusters of neuron cell bodies or ganglia, and they nourish and balance ionic concentrations. Table 10.2 goes through each of these types of neuroglia in more detail. Okay, regeneration. Mature neurons do not divide. If the cell body is injured, the neuron usually dies. Some neuron regeneration occurs in the peripheral nervous system. If a peripheral axon is injured, it can regenerate. Axons are separated from the cell body, and its myelin sheath can degenerate. Schwann cells and neurolemma remain. The remaining Schwann cells provide guiding sheaths for the growing axon. If the growing axon establishes a former connection, function will return. If not, function may be lost. Neuron regeneration in the central nervous system. Central nervous system axons lack neurolemma to act as guiding sheaths. Oligodendrocytes do not proliferate after an injury in the central nervous system, so regeneration is unlikely. Figure 10.10 shows you neuron regeneration in the peripheral nervous system. Cell membrane potential. A cell membrane is usually electrically charged, or what we call polarized. Having charge differences in a local area is called polarity. A charge inside of a cell is called its membrane potential. Inside the membrane is negatively charged with respect to the outside. It results from unequal distribution of ions on the inside and outside of the membrane. It's important in conduction of impulses in neurons and muscle fibers. These excitable cells can rapidly change the internal charge. The ability to change charge allows for communication between cells. So membrane potential is the charge inside a cell. Potential to transport charges across the membrane are what we mean by the membrane potential. Resting membrane potential is a charge inside the cell when it's inactive, and it's about negative 70 millivolts. Potentials result from unequal ion distribution across the membrane. Potassium ions are higher concentration inside than outside the cell. Sodium ions are higher concentration outside than inside the cell. Negativity inside the cell due to mainly the large number of negatively charged impermeate proteins and ions like phosphate and sulfate are inside the cell. Gradients of sodium and potassium ions allow for excitability. Gated channels allow for movement of these ions at certain times. Many chemical and electrical factors affect the opening and closing of the channels. Sodium and potassium ions follow rules of diffusion, moving from higher to lower concentrations across the membrane. Resting membranes are more permeable to potassium than they are sodium. The sodium-potassium pump maintains a balance in ion movement across the membrane. When resting potential is disturbed, it pumps 3 sodium ions out of the cell and 2 potassium ions into the cell. It uses energy of ATP to actively transport these ions in opposite directions across the membrane. Unequal charge distribution and the sodium-potassium pump form the basis for neurons to conduct electrical impulses for communication. An action potential is a sequence of electrical impulses in an excitable cell that involves changes in the membrane potential, first positive, then negative, to return to resting potential. Action potentials are used for communication between the cells. Figure 10.11 shows you ion channels in the cell membrane, a closed one and an open one. Figure 10.12a shows you conditions that lead to the resting potential. So in a hypothetical neuron, before the membrane potential is established, potassium ions diffuse out of the cell faster than sodium ions diffuse in. A net loss of positive charge from the cell results. Okay, figure 10.12, sodium-potassium pump maintains the ion gradient. The membrane potential, negative on the inside of the membrane, aids sodium diffusion into the cell and opposes potassium diffusion out of the cell. As a result, slightly more sodium ions enter the cell than potassium ions leave. However, the sodium-potassium pump balances these movements, maintaining the concentrations of these ions and the resting membrane potential. Figure 10.13a shows a recording of the action potentials. Start with resting potential, you get depolarization, then you get repolarization, hyperpolarization, and back to resting potential. And you may recall this when we talked about muscle contraction changes. So neurons are excitable cells, and they can respond to stimuli. A stimulus is anything that can change the resting potential of negative 70 millivolts in either direction. Neurons detect stimuli and respond by changing their resting potential. An excitatory stimulus opens the chemically gated sodium channels and sodium ions enter the cell. A local potential change is a change in the membrane potential that occurs only in the area of stimulation. Local potential changes are graded. The greater the stimulus intensity, the greater the potential change. An excitatory stimulus opens the chemically gated sodium channels. This makes the inside of a neuron less negative. Subthreshold stimulus does not result in an action potential. Graded stimuli, such as those from neurotransmitters, can add together to produce a threshold stimulus. A threshold stimulus is an excitatory stimulus that causes enough sodium ions to flow into the cell that it reaches. And it reaches the threshold potential of negative 55 millivolts. Now, many voltage gated sodium channels open at the trigger zone, and a charge changes to about positive 30 millivolts. This we call the action potential. Depolarization is the change from negative to positive charges inside the cell, making both sides of the membrane positive. There's an all or none law here. Reaching an action potential either is achieved or it's not. If the action potential is reached, it sends a signal all the way down the axon. Repolarization is a return to the resting potential after the action potential, and it occurs as the potassium channels open and potassium ions rush out of the cell, and then the polarity returns. In hyperpolarization, we have a slight overshoot at the end of repolarization, in which the potential drops below -70 millivolts for a moment before returning to -70 and then to 65. Sodium potassium pumps now return ions to the original locations and concentrations. Okay, and figure 10.13b shows you these changes and the need to hit threshold. Figure 10.14 shows you how ion movements occur during action potentials all along the axon. Okay, table 10.3 summarizes the events that lead to the impulse conduction. So to reiterate, nerve cell membranes maintain a resting potential by diffusion of sodium and potassium down their concentration gradients as the cell pumps them up the gradients. Neurons receive the stimulation, causing local potentials, which may sum to reach threshold. Sodium channels in the trigger zone of the axon open. Sodium ions diffuse inward, depolarizing the membrane. Potassium channels in the membrane then open. Potassium ions diffuse outward, repolarizing the membrane. The resulting action potential causes an electric current that stimulates adjacent portions of the membrane. The action potential propagates along the length of the axon. Okay, there is a refractory period. During an impulse, the portion of the axon actively conducting the action potential is not able to respond to another threshold stimulus. This is the refractory period, and it has two parts. The absolute refractory period. This is the time when the threshold stimulus cannot generate another action potential, and a voltage-gated sodium channeled are briefly unresponsive. The relative refractory period. This is the time when only high-intensity stimuli can generate another action potential. Repolarization is not complete, and the membrane is reestablishing the resting potential. The refractory period limits the number of action potentials generated per second. Okay, impulse conduction. The speed of impulse conduction varies with myelination. Unmyelinated axons conduct impulses over the entire length. Myelin is rich in lipids and prevents water and water-soluble substances like ions from crossing the membrane. It acts as an electrical insulator. Ions can cross the membrane only through gaps in the myelin sheath called the nodes of Ranvier. Myelinated axons transmit impulses through saltatory conduction, in which action potentials jump from node to node down the axon. Saltatory conduction is much faster than impulse conduction in unmyelinated axons. The axon diameter also affects conduction speed. Thick axons transmit faster than thin axons. Thick myelinated axons are about 120 meters per second, where thin unmyelinated axons only move impulses 0.5 meters per second. This figure shows you action potential propagation along the axon. Figure 10.16 shows you saltatory conduction. Okay, clinical applications. Factors that affect impulse conduction. Changes in permeability of axons to certain ions affect impulse conduction. An increase in concentration of potassium in extracellular fluid. This reduces the gradient for potassium to leave the cell. Threshold potential reached with stimulus of lower intensity. It leads to excitable neurons, perhaps convulsions. A decrease in concentration of potassium in the extracellular fluid. Neurons can become hyperpolarized. Action potentials are not generated. A lack of impulses leads to muscle paralysis. And then a decrease in permeability to potassium ions. This can be caused by some anesthetic drugs. It stops impulses from passing through the tissue fluid around the axon. Impulses do not reach the brain, and there's no perception of touch and pain. Let's talk more about the synapse. So neurons communicate with each other at a synapse. This is a site at which a neuron transmits a nerve impulse to another neuron. The presynaptic neuron sends impulses. The postsynaptic neuron receives the impulses. The synaptic cleft separates the two neurons. In synaptic transmission, we have a process by which presynaptic neurons send impulses to postsynaptic neurons. It's a one-way process using neurotransmitters to transfer the message. This figure shows a synapse forming between an axon of a presynaptic neuron and a dendrite or cell body of a postsynaptic neuron. This figure shows the synapse. An impulse is transmitted across the synaptic cleft. As you can see in this picture, we have the vesicles that hold the neurotransmitter, possibly like acetylcholine. And then this is released across synaptic cleft over to the postsynaptic neurotransmitter receptor. Okay, in synaptic transmission, we have transmission of a nerve impulse from one neuron to another. Released neurotransmitters cross the synaptic cleft and react with specific receptors in the membrane of the postsynaptic neuron. The effects of neurotransmitters vary. Some open ion channels, others close ion channels. Chemically gated ion channels respond to neurotransmitters. Local potentials result from changes in chemical gated ion channels are called synaptic potentials. Excitatory neurotransmitters increase permeability to sodium ions and bring membranes closer to threshold, and an increased likelihood of generating an impulse. Inhibitory neurotransmitters move membranes farther from threshold and decreases the likelihood of generating impulses. In excitatory postsynaptic potentials, or EPSPs, the membrane change in which neurotransmitters open sodium channels. This depolarizes the membrane of the postsynaptic neuron as the sodium enters the axon. The action potential in the postsynaptic neuron becomes more likely. An inhibitory postsynaptic potential, the membrane change in which neurotransmitters open potassium channels or chloride channels. It hyperpolarizes membranes of the postsynaptic neuron as potassium leaves the axon. The action potential of the postsynaptic neuron becomes less likely in this case. The EPSPs and IPSPs are added together in a process called summation. Net excitatory effects lead to a great probability of an action potential. A net inhibitory effect does not generate action potentials. Summation of all inputs usually occurs at the trigger zone. Figure 10.19 shows you the synaptic potentials in the summation. There are at least 100 different neurotransmitters. Acetylcholine stimulates skeletal muscle contraction, which we've talked about before. Neurotransmitters may be monoamines, amino acids, or peptides. Neurotransmitters are produced in the rough ER or cytoplasm. When impulses reach the synaptic knob of an axon, neurotransmitters are released by exocytosis. In vesicle trafficking, we have a process of membrane recycling. Synaptic vesicles become part of a cell membrane as it releases the neurotransmitter. Endocytosis returns the membrane to the cytoplasm and forms new vesicles. Table 10.4 goes over several types of neurotransmitters, such as acetylcholine, norepinephrine, dopamine, serotonin, and histamine. And all of these have different impacts dependent on whether they are in the central nervous system or the peripheral nervous system. There's also GABA and glycine, glutamate, enkephalins, endorphins, substance P, and nitric oxide. Table 10.5 shows you several disorders associated with neurotransmitter imbalances, like clinical depression, epilepsy, Huntington disease, hypersomnia, insomnia, mania, Parkinson's disease, schizophrenia, and tardive dyskinesia. Table 10.6 shows drugs that alter neurotransmitter levels. Some of these drugs are legal and some are illegal. So the events leading to neurotransmitter release. So an action potential passes along an axon and over the surface of its synaptic knob. The synaptic knob membrane becomes more permeable to calcium ions, and they diffuse inward. In the presence of calcium ions, synaptic vesicles fuse to the synaptic knob membrane. Synaptic vesicles release their neurotransmitter by exocytosis into the synaptic cleft. Synaptic vesicle membranes become part of the cell membrane, and the added membrane provides material for endocytotic vesicles. Neuropeptides. Many neurons in the brain or spinal cord synthesize neuropeptides. Some neuropeptides act like neurotransmitters. Others act like neuromodulators. The neuromodulators are substances which alter a neuron's response to a neurotransmitter or block the release of a neurotransmitter. Examples, the enkephalins, they relieve pain sensations. The beta endorphins, they relieve pain sensations. They're potent and long-lasting. Substance P is found in neurons that conduct pain impulses. Enkephalins and endorphins inhibit the release of substance P. Okay, clinical application. Opiates. Opiate drugs include morphine, heroin, codeine, and opium. They're potent painkillers, and they're derived from the poppy plant. The human body produces opiates that are called endorphins. Endogenous opiates relieve pain in acupuncture, analgesia during childbirth, and feeling of well-being during exercise. Opiate drugs are very useful in relief from severe pain. Opiates are also very addictive. Withdrawal symptoms, specifically pain, are experienced by addicts that stop taking the drugs, and it's due to the fact that taking repeated doses of opiate drugs cause the body to stop producing its own endorphins. When the drug is stopped, the person is short of endogenous endorphins and therefore feels pain. Impulse processing. This is the way the nervous system processes nerve impulses and acts upon them, reflects the organization of the neurons and axons in the brain and spinal cord. Neuronal pools. These are groups of interneurons that make synaptic connections with each other, and they're located completely within the central nervous system. Cell bodies may be in different parts of the central nervous system. Interneurons work together to perform a common function. Each pool receives input from other neurons, and each pool generates output to other neurons. Pools may affect other pools or peripheral effectors. In facilitation, repeated impulses on an excitatory presynaptic neuron may cause that neuron to release more neurotransmitter in response to a single impulse. This increases the likelihood that postsynaptic cells will reach threshold. Convergence. One neuron receives input from several neurons. Incoming impulses often represent information from different types of sensory receptors. This allows nervous systems to collect, process, and respond to information. It makes it possible for a neuron to sum impulses from different sources. In divergence, we have one neuron sending impulses to several neurons via branching of the axon. It can amplify an impulse. An impulse from a single neuron in the central nervous system may activate several motor units in a skeletal muscle. An impulse from a sensory receptor may reach different regions of the central nervous system for processing. Figure 10.20 shows you a picture or diagram of impulse processing, convergence versus divergence. Okay, clinical application in drug addiction. Drugs alter the activity of a neurotransmitter on a postsynaptic neuron. Either they stop or enhance a synaptic transmission. An antagonist is a drug that binds to a receptor and blocks a neurotransmitter binding. An agonist is a drug that activates a receptor, causing the action potential or aiding in the binding of the neurotransmitter. Repeated use of addictive drugs causes a decrease in its receptors, so the person must take more of the drug to have the same effects. Amphetamines enhance norepinephrine activity, which elevates alertness and mood. Nicotine binds to nicotinic acetylcholine receptors. Channels for positive ions open, which allows influx. And this leads to dopamine release, which leads to pleasurable feelings from smoking. And that is our intro nervous system presentation.